Assalamualaikum and a very good morning to all of you. Alright, for us all to be blessed, Shuriza, please recite the dua.
Bismillahirrahmanirrahim. Allahumma aftah alaina hikmataka wanshur alaina min khazainir rahmatika ya arhamar rahimin. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina azabannar. Wa sallallahu ala sayidina Muhammad wa ala alihi wa sahbihi wasallam. Alhamdulillahirabbil alamin.
The title of our experiment today is Animal Histology, and the objectives as stated in your lab manual are to examine the animal tissue slides under the light compound microscope, to observe and study the structure and organization of the tissue. Before we begin the experiment, I would like to ask you a few questions regarding the animal tissues. I just want to know whether you have done your revision or not before coming to class. Okay, there are 4 types of animal tissues. Name 1. Rosanna.
Epithelial tissue.
Good. Epithelial tissue. The epithelial tissue is further classified into several types. Give me 2 types of the epithelial tissue. Rashida?
Simple squamous and simple columnar, teacher.
Very good. Yes, simple squamous and simple columnar. Apart from that, we also have the simple cuboidal, pseudostratified columnar, and also the stratified epithelium. What are the other 3 tissues apart from the epithelial tissue? Siti Hajar?
Muscle tissue, nerve tissue and connective tissue.
Very good! I am so lucky to be teaching a group of 4 flat students. Look at the slide here. If we take any vertebrates, for example, this bird, you will find all the tissue in the various parts of the animal. The epithelial tissue lines the stomach, the kidney, and also the epidermis. The nerve tissue is in the central nervous system. The connective tissues as the supporting tissues, and the muscle tissues for movement and contraction. Now, apart from naming all the tissues, you should also know how the tissues look like. So that is what we are going to do today. Now please refer to your lab manual exercise 3.1 materials. You won't be doing any slide preparation, which means that you won't have to scrape your precious cheek cells to do this experiment, but you are supplied with the prepared slide for all the tissues in these 2 boxes. You will find the name of the specimen at the end here. Yeah? And after you have finished using the slide, I want you to arrange the slides according to the list, yeah, in each box here. Now look at the procedures and observation. Number 1, examine the slides given under the microscope and observe the structure and organization of the tissues. Draw and label each of the tissue given. Use the appendix below to assist you in your cell investigation. Okay, you will be guided on what to observe based on the diagram on page 16 until page 22. Try to find tissues as illustrated here. The scientific skills that you will gain from this experiment are observing the tissues given, the ability to use and handle the microscope, handling the slides correctly and carefully, ability to sketch scientific drawings. I'm sure by now that you are already familiar with the microscope, so I'm just going to explain it briefly on how to use it. Okay? Okay, first of all, always start with the low power. So in this case, it is the 4 times objective lens. Place the slide on the microscope stage with the specimen directly over the center of the circle on the stage, which means that the specimen is directly over the light. Take the slide and place the slide on the microscope stage with the specimen directly over the center of the circle on the stage. which means that the specimen is directly over the light. Then focus using the cos knob until the specimen is in focus. Then use the fine knob to sharpen the image. Now once you have found the specimen under low power, switch it to the 10 times objective lens. Okay, and then focus again, but remember to use only the fine focus knob. Alright, then again switch it over to 40 times objective lens. Use the fine focus knob to sharpen the image and then draw it. The drawings are required for 400 times only. And before we begin, please put your jotter on the table. I will check them while you are doing your experiment. You may start now. These are the drawings of all the tissues. Remember, you must draw using pencil. Each drawing must be half a page in size and must include clear, proper labels. In the upper left-hand corner of each circle, include the specimen name as written on the slide label. In the upper right-hand corner, include the magnification, in this case 400 times. An unlabeled drawing is nothing more than scratches on a piece of paper. Simple squamous epithelium. Irregular and horizontally flattened shape. Nucleus is centrally located. Cell membrane is very thin. Cell is filled with cytoplasm. 40 times, 100 times, 400 times. Scientific drawings. Simple cuboidal epithelium appear as rings of cells around empty spaces. Cells are roughly square or cuboidal in shape. Each cell has a spherical nucleus in the center. 40 times, 100 times, 400 times. Scientific drawings. Simple columnar epithelium. Cells are elongated and column-shaped. Nuclei are elongated and are usually located near the base of the cells. The cell membranes are very thin but easily identified. 40 times, 100 times, 400 times. Scientific drawings. Simple ciliated columnar. These are simple columnar epithelial cells, but in addition, they possess fine hair-like outgrowth cilia on their free surfaces. 40 times, 100 times, 400 times. Pseudo-stratified ciliated columnar epithelium. The columnar cells of this tissue all begin at the basement membrane. Only a few reach the surface. The nuclei appear at various levels, giving the tissue a stratified appearance. The cilia are easily seen. 40 times, 100 times, 400 times. Scientific drawings. Stratified squamous epithelium. Many layers of cells. The surface cells are flattened. 40 times. 100 times. 400 times. Scientific drawings. Connective tissue, blood, bone, cartilage. Blood. 3 main types. Erythrocytes or red blood cells, leukocytes or white blood cells, thrombocytes or platelets. Bone is made up of osteon or Harvesian system. Each osteon is made up of concentric circles called lamellae around a Harvesian canal. Bone cells called osteoblasts are found in lacunae. 40 times, 100 times, 400 times. Scientific drawings. Cartilage. Smooth, glassy, bluish white appearance. Chondrocytes enclosed in spaces called lacunae, surrounded by capsule. 40 times, 100 times, 400 times. Scientific drawings. Muscle tissue. Striated, smooth, cardiac. Striated muscle. Each cell is cylindrical, unbranched and contains many nuclei. The nuclei are arranged around the periphery of the cell, just beneath the cell membrane. Smooth muscle. Long, narrow, spindle-shaped cells. A single central and elongated nucleus. These cells are arranged parallel to one another. Do not show any striations microscopically. Cardiac muscle. Have a single nucleus centrally located, striated, branched, connected to one another by intercalated disc. Nerve tissue. The functional unit of the central nervous system is the neuron. A neuron consists of a cell body, axons and dendrites. As a summary to this experiment, animal tissues can be classified into 4 types: epithelial tissue, connective tissue, muscle tissue, and nerve tissue. Each tissue has its own characteristic shape and size and serves specific function. For your next experiment is experiment 4, page 23. Introduction to carbohydrate, lipid, protein, and nucleic acid molecules. Now, before we end this session, I would like to remind you that your report must be submitted next week. As usual, the report must contain the title, the objective, the results, the discussion, and also the conclusion. Emphasize on the structure and function of each tissue in your discussion. Yes. So that is all for today. Yes. Thank you, and I will see you in the next experiment.